A common insect sometimes found around a house is the fruit fly, Drosophila. Fruit flies are very small compared to an ordinary housefly. Adult fruit flies are only about 3 millimeters long. In a laboratory, we can anesthetize some fruit flies and examine them closely. After a short time, they can be removed from the bottle. With magnification, we can see their red eyes and transparent wings. Because of their short life cycle, many generations can be produced in a short time. So fruit flies have become a favorite laboratory animal. They are widely used in experiments concerned with genetics because of their short life cycle and large chromosomes. They are also used for other kinds of studies. We're going to perform some experiments to help us learn something about fruit flies and their environment. These 3 containers look the same, but let's compare the activity of the fruit flies in each box. In the middle box, the flies are quite active. In the left-hand container, there seems to be less activity. Do you think the conditions in the boxes are identical? What might cause differences in activity? The flies in the middle box also seem more active than those on the right. When we remove the board, we can see that the containers are kept at different temperatures. In the box on the left, being kept cold, very few of the flies are moving. In the container that is heated, we can see almost no motion. What about the middle container? It's at room temperature and seems to provide the best environment for the flies. But room temperatures may vary. Let's see if we can determine at precisely what temperatures the fruit flies are most active. This flask contains a centigrade thermometer. Now the flies are still active. But watch. Read the temperature and see if the activity of the flies changes. 20 degrees centigrade is 68 degrees Fahrenheit. 10 degrees is 50 degrees Fahrenheit. Below 10 degrees centigrade, the motion slows down. Are most of the flies dying? We can find out by placing them under a heat lamp. Watch the flies as the temperature goes up. At what temperatures do they seem most active? What happens as it approaches 25 degrees centigrade, or 77 degrees Fahrenheit? And what happens when the temperature goes even higher? After experimenting with a large number of flies in this way, we can graph our results. When temperatures fall below 20 or rise above 30, the fruit flies become increasingly inactive. Between 20 and 30 degrees, they are most active. If we breed some fruit flies, we can learn more about how they respond to temperature. These flies have been anesthetized. Now we can sort them to select the ones we want for the breeding experiment. One male and one female are chosen. The male has a dark-tipped abdomen and is thinner than the female. The female's abdomen is thicker and more pointed. The selected pair is placed in a container along with a food medium. After about 2 weeks, the bottle is swarming with flies at different stages of development. Inside, we can see mature flies, plus eggs, larvae, and pupae. Let's remove some of the food material from the bottom of the bottle. We can examine each stage of the fruit fly's life cycle more closely. These are eggs beginning to hatch into larvae. Each egg is about 1 millimeter long. These are newly hatched larvae. Each larva grows to be about 4 millimeters long. In addition to the larvae and the eggs, we can also find pupae. Some of the pupae are ready to hatch. In this dish, we have a newly formed pupa. Let's remove the outer covering, the pupal case, and watch its development. With time-lapse photography, over a period of about 3 to 4 days, we can watch the pupa developing into an adult fly. Now, watch the development of the head and the large compound eye. Here we see the newly formed adult coming out of the pupal case. The wings are not yet fully spread or dried, but the life cycle has been completed once again. Now let's experiment with controlled breeding. This is a temperature control apparatus. Each of the knobs is set for a different temperature and connected to a breeding tube. Each centigrade thermometer shows a different temperature which remains constant. Now, male and female flies are placed in each breeding tube. Which temperature do you suppose will provide the best environment for the flies? We'll watch the tubes over a 2-week period to see what effect temperature has on reproduction. As the days pass, we can see that the flies are multiplying. Now we see how much temperature influences the reproductive rate. Is the best temperature for reproduction within the range of temperatures we found best for general life activities? Now let's try one more demonstration of reproduction and see if you can explain what happens. Right now, the temperature of the air and the temperature of the water are the same. We'll leave one breeding tube out in the air and place another in the water. What will happen? We'll wait for 2 weeks. Consider what changes might occur in the temperature of the air and the temperature of the water during the 14 days and nights. At the end of the 2 weeks, we compare the tube from the air with the one from the water. Are there differences in the rate of reproduction? Can you account for the results in terms of what we saw about the fruit fly and relationships to its environment? What do such relationships suggest for other organisms and their environments?